Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. My guest this week is Dr. José Leme. Dr. Leme graduated with his bachelor's degree in agronomic engineering from Universidad Estadual de Londrina. For his master's degree, he studied precision agriculture at Auburn University, followed by his PhD at Virginia Tech with a focus on plant sciences. He started at Southern Illinois University in fall 2021, bringing his expertise in cannabis science and indoor cultivation systems. He has a joint appointment in the School of Forestry and Horticulture and the School of Biological Sciences. He is the principal investigator in the Cannabis Biology and Cultivation Systems Labs, focusing on areas of controlled environment agriculture, plant secondary metabolites, nutraceutical pharmaceutical biology, phenomics, plant physiology, biotic-abiotic stress, crop steering, and growing practices. His team aims to determine how to leverage environmental controls, plant nutrition, biostimulants, and hormones to improve plant production. Dr. Leme will be speaking at the Cannabis Cultivation and Science Conference this September 5th through 7th, and tickets are still available. You can learn more about this event on the Kiss Organics website. This is going to be the educational and networking event of the year, and it includes a facility tour of Collective Elevation, 2 days of amazing speakers, and a third day with an included tasting event sponsored by the Montana Living Soil Alliance. I'm looking forward to seeing people there. It's coming up fast. Now on to the show. All right, Dr. Leme, welcome to the show.
Thank you. I'm happy to be here. Finally.
Yeah, it's great. I'm glad we got to connect. Can you tell me a little bit more about yourself and about your programs?
So yeah, I'm assistant professor of cannabis biology and controlled environment agriculture here at Southern Illinois University, and I have a split appointment in the School of Forestry and Horticulture and the School of Biological Science. So I have 51% in horticulture, 49% in plant biology. So our lab, it's very diverse, and I would say in terms of not only nationalities and genders, but also scientific mindset. But basically, we're working with indoor cultivation of plants and testing different abiotic, biotic stressors, environmental controls to improve cannabis quality and production. Because I think differently than other plants cultivated indoors, for example, like food crops like tomatoes, cucumbers, and basil, that the end goal is pretty much biomass or fresh fruit. When you're producing cannabis, it's a fine line, or a balance between production of biomass and secondary metabolites. So when you're producing biomass, you're prioritizing optimal conditions. But when you're producing plant secondary metabolite, stress or a stimulus that is kind of controversial. Some people don't really like to talk about to use the term stress, especially in crop steering, because they see more as a stimulus. It's not actually detrimental for the plant. So then when you're producing cannabis, it's unique because you're producing biomass, but also secondary metabolites. So, and that's pretty much one of your main goals in our lab here at SIU.
Yes, so you're at Southern Illinois University. You brought up a really good point, I think, in perspective I haven't really thought about. But when we're growing a traditional crop, we're growing it for the fruit. We're looking to eat it. But when we're growing cannabis, yes, we want biomass, but the only reason that we want that biomass is to increase the concentration of secondary metabolites. So essentially to get higher concentrations of THC and cannabinoids and terpenes associated with cannabis flower or inflorescence, as it's called in the research community. Mm-hmm. And you talk about stress. I did a really interesting podcast on plant stress. And I think it's a really fascinating topic because, as you know, and as you stated, secondary metabolite production, so the production of THC and various cannabinoids, is highly linked to plant defense as a plant defense mechanism, but also plant stress. And there's a variety of different things that can fall under plant stress. Do you want to kind of talk a little bit about that?
Yes, yes. And the thing is, even the term stress, not more recently, I'm realized that it can be controversial. For me, it was not really controversial, but yeah, I'm seeing, especially in the cannabis scene, I'm seeing a lot of people that don't like to use the term stress, especially because when you're trying to use the crop steering approach, you're seeing it as a it's just a stimulus because you're not actually stressing. It's just a trigger to increase the production of plant secondary metabolites, and then it's not really being detrimental for the plant. For the studies, for the publications that we might talk later in this podcast, I for most of them I decide to use the term stress because we actually used extreme water depth stress. So it was not just a stimulus. And then again, it's hard to know when it's just a stimulus and not a stress. So but yes, as you mentioned, like when you're producing cannabis, it's a balance between the biomass production and the production of plant secondary metabolites. So um, uh, and again, that's why it's important to have that trigger and not just promote the optimal conditions for the plant, because of course, naturally the plant will produce plant secondary metabolite, but if the plant is not under stress, and it can be water depth stress, temperature stress, humidity stress, uh, nutrient deficiency, light intensity, different light spectrum, light recipes. So, and even mechanical stress that we might talk as well. It can trigger the production of higher production of secondary metabolite. Like a good example that I like to use is like adrenaline. Like secondary metabolite is, as the term is saying, is not primary. So the plant can survive without the plant secondary metabolite. Uh, and it's like adrenaline. If your body is not under stress or like a weird condition, you're not producing adrenaline. And it's the same thing for plant. If the plant is if you're not creating a stimulus or a trigger, the plant is not going to increase the production of secondary metabolites. But again, but it's hard to find the right spot of stimulus, trigger, or even stress.
Yeah, so essentially, stress comes in a lot of different ways, or stimulus, either term here. And you mentioned a bunch of different, abiotic stress or triggers like environment, humidity, temperature, light, spectrum, watering, moisture, water access is a huge one. I'd add herbivory, pest pressure and pest stress, all of those things. And I guess for me, one thing that I've kind of said for years on this podcast, and maybe that will change as research improves, is that just through the production of a plant in a facility or in your home grow, you probably have multiple stress events occurring or multiple stimulus happening that are going to trigger secondary metabolite production. And a lot of the intentional stress stuff that we see, or even unintentional, let's say we forget to water or our lights are too high, a lot of those things will have a dramatic impact on biomass production or our overall yield. So if we have too much spider mite pressure, or our light intensity is too high relative to our environment, or I don't know, fertility, if we don't give the plant the right amount of fertility, we're going to see we're going to see increased secondary metabolite production, but we're going to see a dramatic decrease in yield
Hmm.
potentially too. So, like you said, it's really about getting that optimal balance there. And I don't know that we've, one thing I've said on here, and maybe this is no longer true, is that I don't know that we've figured out, especially with the genetic diversity in cannabis, where those optimal trigger points or stress points are, where we could say, hey, week 3, we want to do a water restriction, or we want to, raise CO2, or all of these different things to really optimize it from a stress response, secondary metabolite production perspective. I don't know. What do you think about that?
100%, 100%. Because also it's so many moving parts. Because it's not
It
only about
is.
one stress. Because like as we mentioned, like light intensity. Okay, you can if you increasing light intensity, but you're not increasing CO2, you create an imbalance. So you're creating a stress. So, and if you increase CO2 and light intensity, uh, also another factor that can be affecting is the spectrum. Um, so talking about water deficit stress or stimulus, or so the dry back that we talk in crop steering. Like, what is the magnitude of that stress? Is like you're pretty much drying that entire rock roll cube, or the entire pot, or it's you're letting the pot to with a little bit of water, or for how many days is gonna last that stress. What is the best growth stage? Is early flower, late flower, mid flower? And again, as you also mentioned, the genetics. Is that genetic very responsive to that specific stress? Um, so it's so many moving parts, and then it's hard. That's why I can see different results out there in terms of what is good, what is bad, if it's beneficial, if it's not beneficial, if it's changing anything, or if it's just losing biomass and it's not gaining any, uh, increasing, cannabinoid concentration. So yeah, I agree 100%. It's so many moving parts, and to find, a specific answer, it's not easy.
Yeah. So what I'm getting is stress, or these stimulus triggers will cause an increase in secondary metabolite production, but they will potentially reduce yields. We don't have it all figured out, and it's very complicated and probably tied more specifically right now to the genetics that you're running. It's not necessarily going to be consistent across the board, so we can't make broad sweeping claims about all cannabis plants. And hopefully we'll just continue to see more research on it. Now, you recently published a paper in June of this year on the effect, I'll just read the title, the effect of water deficit stress on cannabis sativa development and production of secondary metabolites, a review. And I think this paper is really interesting. I don't want to give away all of it because you're going to be presenting at our upcoming Cannabis Cultivation and Science Conference specifically about this paper. So I want to keep some of it, I want to keep some of it, some of the surprises for that. But can you give us a little overview of kind of what you were looking to achieve, and a little bit about what your findings were maybe?
Yes, yeah. Actually, this paper is really cool. This project was conducted by one of my master's degree student, that Shiksha Sharma. She just graduated. Now she's in at the University of Tennessee, and that review is actually her literature review. That you pretty much she's pretty much summarizing, or is an introduction of the 2 other chapters. It's the first chapter of a 3-chapter master thesis. So she's just introducing the concept, and it's pretty cool, because the first time she did the experiment, we stressed the plants for only 5 to 7 days, and then when we ran the analysis, it came back all not significant, not statistically significant at all. And
So you
then
you didn't water, you didn't give the plants access to water for up to 7 days, and you didn't see any difference.
no, the plants were
Wow.
coming back like as nothing was happening. So, and like no statistically significant result in terms of biomass and cannabinoid or in terpene secondary metabolites in general. And and then I talked to her, and she was like, "Well, I mean, I want to publish something, like with these results, it's not much I can publish because it's nothing significant." So then we decide to um, create or promote a very extreme water deficit stress. So I was like, okay, because she was also worried about killing the plants. So and this is actually a two-part study. So the first study, we are trying to find the best frequency of water deficit stress during the flowering stage. So the first study we test control no water stress like regular irrigation, 1 period of water stress, 2 period of water deficit stress, and 3 period of water deficit stress. So then after this study, finding the best frequency, we want to find the best timing in the growing stage. Because again, I'm not going to give much away, but during, and again, that's going to be my presentation at the conference. Throughout this study, but before getting the cannabinoid analysis. We had the biomass, we had the trichome density counting, main inflorescence weight. Throughout that, when we got all that results, we kind of realized that 1 period of water depth stress was the optimal because we were not losing total and bucket biomass, and we had the trichome density was increasing. And I was like, okay, this is promising. Biomass, not statistically significant different when compared to the control, and we're increasing statistically significant, higher trichome density. At that point, I was like, okay, this is promising. We can start the timing study. But then you see that later everything changed, and we'll talk about that in the conference. But yeah, so then the second study, we had the study is pretty much different timings, like no water deficit stress, early flowering, middle flowering, and then late flowering. Also, actually, we have actually 5 treatments: control, early flower, mid flower, late flower without rewatering, and late flower without with rewatering. So we are kind of giving some time to the plant to recover from that stress. So that's the second study that we conducted. We are also work on this data.
Now, just for listeners,
Mm-hmm.
so they can better understand the framework of your methodology, what media were you growing in, and what were you using for nutrition?
So yeah, across the board, we use a regular standard fertility liquid fertilizer. We use peat and perlite as a media. We try to
Okay.
be, to use a inert. No, we are not using living soil because when you're dealing with living soil, it's really complicated.
Yes.
And also we are not using rock roll because it's so um, quick. It changed so quickly. Of course it's awesome for crop steering, but when you're conducting a study with many students that are helping, sometimes it's too quick. So peat perlite is a good media for us. It's still inert, and so yeah, we use a standard uh, fertilizer regime for veg and flowering across the board for all the pots. And so then pretty much the only different where the water deficit stress, which is also consequently nutrient deficit stress, because when you're not adding, when you're not fertigating the plant, we are also not adding, nutrients. You're not feeding the plant.
Yeah, they don't have access to nutrients if there's no water. And
Mhm.
as that water is drying out, you're getting an increase in electrical conductivity, in EC in that media as well, which also can be a stressor. Good point. So the
Surprisingly,
other thing,
pH didn't change that much throughout the study.
and you're using a mineral salt or chemical fertilizer as well for this.
Yes, yeah,
Just to
in
clarify.
balance, like pH balance fertilizer. Yes.
Yeah, no, I would add that most research that we're going to see on any crop, but with cannabis as well, is going to be a similar, either using rock wool or an inert media like peat perlite and mineral salts, because of all the complications and variability in living soils. It's just
Hmm.
much harder for a scientist to draw any sort of statistically significant conclusion because you can't you can't remove all that variability. So, as someone who grows in living soil, I have to take this research and then start figuring out how to apply it to what we're doing and what I'm doing in the organic living soil world. So, yeah, I just wanted to share that. But let's also touch on the fact that, because the thing I was thinking about with your media being peat perlite, that's going to hold on to the physical properties of it. It's going to hold on to water better, I would assume, than say rock wool, but maybe not as good as something like living soil, where you have
Hmm.
more biological activity. That's also, these microbes are going to hold water within their cellular bodies. So how often were you watering, I guess, in your peat perlite mix approximately? And I realize that's going to vary during the flower cycle, before you did these drybacks.
So well, for the control, we are keeping the water. It had like a volumetric sensor.
Yeah.
So we are keeping the soil, the volumetric level in the pot between 30 to 40%. So when we're reaching that range, 30 to 40%, we're adding water again for the controls as a regular fertigation regime. And then for the treatments for the plants going under the water that's stress, then as soon as we reach that flowering stage that we want, or the frequency that you have in the protocol, we are just stop irrigating for pain. So then, after the first study that we test for 5 to 7 days, the second study we actually stress the plant for 10 to 12 days. No water,
Wow.
zero. Yeah, and the plant survived. The plant survived. And again, something that I told my student was like, okay, if all the results are not good, or if they don't know what we're doing is not that valuable, at least we are learning that if you're a home grower and you want to travel for like 2 weeks, let's say, and you have nobody to take care of your plants, you can just go, come back after 12 days, rewater the plant, and the plants will survive. At least survive.
Yeah, yeah. I mean, I think that's going to be somewhat dependent on evapotranspiration rates, like your VPD. If you're running really high temperature, higher humidity, I would assume that you're going to dry out even, or see plant stress sooner than a
Mhm.
plant in a cooler, less humid environment. But yeah, I think that's really interesting, and I think we do need failed experiments, because that teaches us sometimes as much or more as successful ones.
Yes, yeah. I mean, VPD was not a parameter, so we kept the VPD and the other, growing parameters equal for all and optimal for all,
Yeah.
including control. So, but yeah, if you're not really regulating your VPD, and a VPD, and the transpiration is pretty high, yes, maybe 7 days will be detrimental, and maybe if you come back after a week trip, your plant will be die. But if you're controlling the VPD, which is possible to do it, online, or you don't need to be there to control the VPD, it's again, it's you we might be able to come back and your plant be it's alive.
Yeah, when I think about when I screw up and have VPD swings, or forget to water, and the soil gets too dry, I just assume I'm increasing secondary metabolite production. So I try and spin it positively, but yeah, it is potentially hurting the yield.
Yes.
Cool. Well, I'm excited to hear about those results. I don't want to spend too much more time on it because you have a bunch of other research coming out of your lab that I think is really interesting. But I'm looking forward to seeing you at the conference and hearing about that.
Yes, I'm excited too.
All right. So this next paper that you sent me contained a word I'd never heard of before, but once I read the paper, I could understand what you were saying. The word is thigmomorphogenesis. Do you want to talk about what thigmomorphogenesis is, what it means?
Yeah, this is also a pretty cool project that is a collaboration with a company from North Carolina, Glenn Holland. He's a great supporter. And so basically, thigmomorphogenesis, the first time that this term appeared in the literature were in 1973, something like that. And the description of thigmomorphogenesis is the effect of mechanical stress in plants. It can be like a wind, it can be a bug that is flying around, the plant, any type of mechanical stress, or even like a damage, mechanical damage. So that under this description of thigmomorphogenesis, we decide to create a study where we are testing vibration, like mechanical vibration, and supercropping, um, as a mechanical stress. Um,
High
so
stress training, I
high
think, is
stress
how you
training, and
listed the
super,
paper.
yeah,
Yeah.
yeah. Some people know as a high stress training. Some people know as a super cropping. Um, so again, both are mechanical stressors, and high stress training is actually it's a common practice in commercial facilities, but
Yes.
there's not much studies proving that if it's beneficial, if it's not beneficial. So then we decide to test this both mechanical stress. Another interesting thing about mechanical stress is, I mean, people talk a lot about sound. People plant how sound can affect plants, and but again, sound, the sound waves is still a mechanical stress or mechanical
Stimulus.
influence,
Yeah.
but it's minor, it's really minor. So it's really hard when you have students that want to publish something, and then start testing sounds. So then when you're vibrating the plant, actually vibrating the plant, this is something more effective, I would say. And so, and in this study, again, it's not published yet. We're still working on it. We just got the cannabinoid analysis, and we are working in the statistics. But yeah, we had four treatments, control, only vibration, and the vibration was promoted by a grid that, with a transducer that promoted like a 200 hertz and 15 decibels, 15 minutes per hour during the entire cycle of the plant. So the other treatment was super cropping or high stress training, and the fourth treatment was the combination of both uh, vibration and super cropping. So far, the results we have, we clearly could see some morphological differences. And of course, when you're super cropping or high stress training, you're breaking that apical dominance. So
Yep.
it's really changing the morphology of the plant. The plant is growing more like a reverse umbrella instead of like Christmas tree. Um, but in terms of secondary metabolites, we don't have the results yet.
Okay.
But we are, looking for to see if these stressors, mechanical stressors, are changing, the production of secondary metabolites.
Yeah. So just to back up, when we talk about mechanical stimulation, just for folks that are listening, there's a variety of different things that would qualify for that. Like you mentioned, wind. So in an indoor facility, having a fan that's lightly moving the branches back and forth, we know that will increase it will stimulate some root production to help hold the plant in place, as well as increase the thickness of the stalk, I believe.
Hmm.
So the plant will not fall over, essentially. If you have a plant that gets no stimulation whatsoever in that form, a lot of times you have weaker stalks and it can sometimes fall over. This is just something I know from coming out of the nursery in landscape industry growing up when we plant trees. Other forms of mechanical stimulation would also include pruning. When we prune a plant, we're
Yes.
wounding it, and that stimulates it. If an insect like a spider mite punctures that leaf, that's called herbivory. That's also a mechanical stimulation. Um, let's see,
Low
what else
stress
did you,
training. Low
low
stress
stress
training.
training?
Yeah,
Yes,
another one. Yes.
LSTs, another. So
Yeah.
all of these things fall under this thigomorphogenesis.
Yes.
Such a mouthful.
Yeah.
Just for folks who are thinking about it, and we know that like these things will increase secondary metabolite production for the most part. Like there's research to show that with, I believe, other crops and other plants, and there might even be some with cannabis. I'm not sure. What surprised me though is that no one has looked at super cropping or high stress training on
Yes.
the university side, because it's such an accepted practice that
Hmm.
knowing that we want an even canopy in indoor facilities so that we can get better yields is fairly well accepted at this point. That uniformity of canopy leads to much higher yield production. I guess I would say anecdotally, if there's no research around it, but it's fairly accepted practice. What did you find from your study? I would think that at the very least, high stress training by itself would beat the control. Um, did the vibration by itself or in conjunction with high stress training really make a difference?
So, in terms of biomass, we
Yeah.
did not find any statistically significant results, all the same
Really?
biomass. But again, we found numerically differences, but the statistics didn't catch it. So, but again, numerically is numerically, sometimes it can affect, the financial impact,
Yeah.
like if, the stats might say no, all these treatments are equal, but if you have a numerical difference, you can, the finances can be impacted. Um, what we found, I mean, I have to, I don't remember exactly, but we also found a difference in a person, again, numerically person, the difference in terms of biomass stem ratio among the treatments.
Okay.
And and if I'm not mistaken, I think the
I have it in front of
do you
me
have it there?
actually.
I think the
Yeah.
combination of all, the combination of high stress training and vibration had the highest uh, uh, biomass stem ratio, right? I think 3 or 4%.
Yeah, so you list the control at 71.89% biomass,
Hmm.
whereas the high stress training and vibration came in at 74.54% biomass. So what you're saying is essentially, yeah, that's about let's just call it a 3% increase, which
Hmm.
in a commercial facility, 3% could be the difference between profitability and
Yeah.
failure. I mean, 3% of a crop can be a dramatic amount of income.
Hmm.
But for your experiment, you run a series of mathematical statistical equations, and those may show you that that difference is not considered statistically significant by the scientific community based on the standards that everyone uses.
Yes. Yeah. 95% confidence.
Yeah, yeah, yes.
Sometimes you use 90%, but yeah, the scientific community accept 95% confidence.
Yeah, I would want to dive like really dig into this experiment and find out like how full that canopy was across the board, because I would assume that the high stress training in conjunction with an optimal veg cycle to fill that canopy and give you a more even canopy would show an increase
Mhm.
over a control where the plant's growing with, strong apical dominance.
No, yes, and also, as we conduct that in a controlled environment growth units, that can affect as well. If you're doing that like experiment in a indoor facility with
Mm-hmm.
more plants, because then we have more space to open the canopy and actually have, instead having light competition, we will have more light exposure. you
Yep.
know and so, but that's are some of the limitations that we have, in our research, or in our research settings. But another thing that was actually very interesting is that it's well known in the literature that mechanical vibration can decrease inflorescence elongation. There are many studies showing that, and we found that because the treatment under vibration, only vibration, presented the heaviest cola, the heaviest main inflorescence. So, and our assumption is that as vibration, mechanical vibration, is decreasing the main inflorescence, you're creating a more compact, more compact cola, or heavier cola, because you're not actually elongating the cola. So in that same size, you know, uh, 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 cola, it's a heavier one. It's increasing the weight. So that was actually an interesting result too.
So denser buds with vibration, essentially,
Yes,
is what you're saying.
yes, yes.
That's interesting, and I think you bring up a good point that we need to take these studies and really, rather than just saying, drawing any strong conclusions. Like, I can't look at this and now say, okay, high stress training doesn't work because Dr. Leme did this trial and the biomass results, the yields were the same. I think we have to dive into them as someone who's reading a study and say, okay, well, maybe we need to look at like how many A quality buds versus B quality buds did we get between these two, and experiment with different genetics and veg times. And so what I think these studies show us though is like, okay, this is really interesting to me. Now I want to do a trial incorporating some type of vibration, to see if I see in my environment an increase in yields or an increase in cola or bud density. That would be really interesting. So I think studies like this really spark innovation and give us things to trial as growers in our environment or in our facility.
But also, that's the price we pay when you're conducting research in cannabis. Like, we are being a pioneer. Like, we are just, it's the first studies,
Yeah.
and it's funny that that specific study that you mentioned about the water deficit stress, when I post about it, something about it, somebody comment, "Oh, Kaplan," and I know we cited that paper. Kaplan from Guelph University of Guelph, already
Yeah.
did this study, and I was like, "So what?" Like,
Yeah.
for other plants, people are redoing so many, studies, just tweaking the protocol a little bit. So why we cannot do that in cannabis? It doesn't mean that if you conduct one study and it's done deal, you know.
Yeah, that's a good point. We need multiple studies and we need multiple replications
Mhm.
of studies to really form any sort of like scientific consensus about this plant. Because especially in this crop compared to other traditional crops, we have way more genetic diversity. Like, it's not
Yes.
even close. In corn, we have just a few varieties that most people, few cultivars that most people are
Yes.
growing. We'll talk terminology here in a second. Whereas in cannabis, we have, I don't even know, thousands of cultivars probably. I mean, the genetic diversity is massive. So I would expect different results with different cultivars, just based on that genetic diversity. But maybe over time
Exactly.
we'll learn. Yeah, maybe over time we'll learn that OG Kush grows best using stimulation and high stress training, whereas I don't know, cookies grows best using very little mechanical stimulation whatsoever. It really, I think, could be cultivar specific down the road.
Not only cultivar specific, but also the rest of the growing environment, light spectrum, fertilization recipe,
Yeah,
fertigate, water, scheduling, how much water you're adding. So there are so many other moving parts that also can affect the final result. So, and for these other plants, we have 100 years. People are studying and changing and learning more, and changing their mind and redirecting the research. But for cannabis, it's just the beginning, so we don't have much background information. So again, that's why my students are struggling to write their literature review, because it's like, hey, we don't have much papers. The paper,
yeah,
and now we have recently, it's getting better, but it's not like if you're writing a literature review for soybean. You know,
yeah.
so
There's too many papers. Yeah,
yes,
yeah.
exactly.
So, real quick before we move on, it sounds like your 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 potential conclusions or findings with this study is that while the yields did not change across all the different treatments or variables on this, you did see an increase potentially in secondary metabolite and terpene production. So essentially, higher levels of cannabinoids and terpenes from this thigmomorphogenesis, this mechanical stimulus.
Yeah, I guess our assumption or hypothesis, like, in
There we go.
as far as we know, as biomass was not affected, statistically not significant. If we have a higher cannabinoid concentration or higher terpene concentration, plant secondary metabolites in general, it will be equal to higher productivity, because same biomass, you're not actually losing biomass due to the stress. So, because the other result could be, okay, these stressors are actually decreasing biomass. So if my cannabinoid increase a little bit, it's going to be equal to the plant under optimal condition or non-stress condition, because the biomass was higher. And then when you're getting the total biomass, total cannabinoid per plant, it's end up being equal. So that's why it's the balance, biomass and plant secondary metabolites.
So our goal with any of these stress or stimuli is to increase trichome density without dramatically lowering biomass production or yield. Because if we have more trichomes being produced, then in theory we'll have higher levels of cannabinoids in the same amount of biomass. That in the sense what we really are going for here are cannabinoids when we grow cannabis. That'd really be considered a win, I suppose.
So,
That's the
well,
goal.
okay. So what is your opinion about trichome density? Do you think higher trichome density is directly correlated to higher cannabinoid concentration?
Well, and correct me if I'm wrong, but my understanding is if I have higher trichome density, I may not have higher levels of THC necessarily, but I'd be harvesting something that would have more overall THC and cannabinoids than something with lower trichome density. Is that a wrong assumption?
Well, I mean, the thing is, that's the general assumption. And the thing
Okay.
is, people actually even get mad when, mainly legacy growers, they get mad when you do not agree with that. But unfortunately, or trichome density counting is not being directly correlated to cannabinoid concentration. And this is something that already I've been in a panel with Dr. Alison Justice, with Dr. Nirit, and we are kind of finding the same results. And the thing is, and this is something that I addressed in my very last publication. Or I think we need to improve our trichome density counting methods. Because the thing
Okay.
is, let's say when you're during the trichome density counting, you cannot count all the trichomes, in a flower. So you take a representative part of the plant and extrapolate that to the rest of the plant. So then with that, the first issue is what inflorescence is structure you're gonna be using as a representative part.
Sure.
The
What part of the
bract,
flower
yeah,
are we
what
going to
part of the
count?
flower? The bract, the entire flower, just a random spot in the inflorescence, the sugar leaf, the fan leaf. So we have people are using different, plant structure, and then so that's the first issue. And then again, the fan leaf can have higher trichome density than the bract, or the bract can have a higher
Really?
trichome density than the fan leaf. So and then when you correlated that density with the cannabinoid concentration is not being directly correlated. Another issue is when you're counting trichomes, you're counting basically numbers. You're not counting the size of the trichome head. How
Mm,
tall is the trichome? So when you're counting numbers, let's say 5 big trichome heads could be equal to 10 small trichome heads, but what the trichome density is showing that 10 trichomes is higher than 5 trichomes,
yeah.
that's why that correlation, we're not really finding that correlation. And so that's why we need to improve our methodology to be more representative or
I see.
somehow, you know, correlate to the cannabinoid levels.
Would you agree, though, that the same genetic, same cultivar with higher trichome density should, in theory, have more cannabinoids and more THC than the exact same plant with lower trichome density, or does that not even hold true?
I would not make that statement based
Okay.
on what I'm seeing.
We're still learning that. Wow, that's really interesting. So
Yeah.
trichome density may not really matter is essentially what you're saying. We don't know.
No, I think it's a very important metric. I think it's a very important parameter. But basically for aesthetics, because the thing is, one thing you cannot lie: if you have a higher trichome density, you have a frostier bud. That's for
Yeah.
sure.
Yeah.
But
That's what we look for as growers and as
yes,
consumers. People see that and people want those genetics just because of the trichome density, how frosty it looks, as you said. Yeah.
that's why it's still a very important parameter because it's a quality parameter. But to correlate it with that with cannabinoid, again, you also need to consider the size of the trichome heads. It's also what part of the plant you're using as a representative uh, 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 sample. We use here in our lab, we use the bract because so far we're seeing that the bract is the most homogeneous structure.
Okay.
So, and again, we cannot be all day counting trichomes, counting the entire, all the trichomes. So another thing that I'm suggesting that paper is to use more than one part of the plant: the bract, the fan leaf, the sugar leaf, and
Yeah.
then you can kind of create an average of all.
We do the same thing in the microscope world. It's called MPN, and essentially it's an equation. So we'll have a slide that's broken up into this looks like graph paper, let's say, and then you count essentially, a certain number of the little squares, and then you take that and extrapolate how much bacteria you have based on a math equation, and it gives you a reasonably good idea of where you're at, in terms of microbial biomass, because you can't count all the bacteria, and the same thing here, like you're describing with trichomes. So that's really interesting. Yeah,
And
so I
and
guess
and,
that's a
and we're
good
using
assumption.
yeah, and we're
Not to
using a 1 millimeter square, and then in 1 millimeter square we can count from like 5 to 30 trichomes. So it really range. It's a big range. So, and again, totally different size in terms of globular trichomes head. So
Yeah.
again, and again, going back to the genetics, we always go back to the genetic. Some genetics might produce more trichomes in the fan leaves or in the sugar leaves than in the brackets. Some genetics will be create more trichome in the bracket. So you cannot just generalize for all the genetics.
Yeah, and I think just to further complicate things, I don't know that trichome density is a good tool for evaluating the cannabis experience or the
Mm-hmm.
user experience in terms from either a medical or a recreational perspective. I think we have to look, much closer at cannabinoids and terpenes and what people call the entourage effect in really evaluating cannabis quality. But unfortunately, right now, at least in the commercial market, it's really tied to trichome density, visual aesthetic appeal, like you
Mm-hmm.
said, and it's tied to THC levels in most what I would call immature markets, recreational or medical markets. They just want the highest level of THC because they think that's best. And if you talk to anyone who's been in the industry or is a much heavier consumer, they will tell you that that has very little impact. Like it's not always about chasing the highest THC level. It's about finding that entourage effect that really gives you the best consumer experience.
Yes, yes. And you definitely see more and more the the threshold is the THC level, but people are seeing more and more that that's not what matters. The
Yeah.
entire all the terpenes, the entourage effect, that what matters in terms of experience. So, and another thing that is well known in talking about trichomes, is how tall is the trichome, because as the trichome grows, it creates more cells to produce energy for this little biofactory that is in the trichome head to produce secondary metabolites. So again, and when you're counting trichomes, that how tall is the trichome is also not being taken into consideration. So that's another factor. That's why you're seeing this kind of the correlation is not being directly uh, uh, correlated to cannabinoid concentration.
Yeah. Now, you didn't send me any research on this, but I feel like users or listeners will be upset with me if I don't ask you. In terms of tricho maturity, do you have any information or research? Because, people say, you want it to be, cloudy versus amber, and, versus clear. Like there's a lot of different opinions on that. Most people I know want the majority of the trichome heads to be cloudy with some amber in there. Have you seen any research on trichome maturity in reference to secondary metabolite production or optimization?
I mean, surprisingly, there is only one study that I found that is exactly, they're actually studying cloudy, amber, and crystal trichome. So far, I just found one study, and it's actually kind of old study. I think it was done in the 90s,
Wow.
and that study was literally taking a needle and measuring the cannabinoid concentration in a very cloudy, amber, and crystal trichomes. And in that study, it was found that the cloudy trichomes, it was higher cannabinoid concentration. So, and there are other studies not being that specific, but they're showing that when the trichome is becoming amber, you're degrading, so you're losing cannabinoid. So, uh, so that's why I would say, in my opinion, so far, it's the cloudy trichome is the best harvest time. You know, because that's another issue. Okay, when should we harvest? Should we just do like a automatic or regular schedule? Okay, after 8 weeks, 9 weeks, 10 weeks, we harvest, doesn't matter? Or should we check the trichome color? Should we check other parameters? So that's another thing that we need research, you know.
Yeah, I do know that or I've heard, I guess I shouldn't say I know anything here, because this is not an area that I have as much interest as I do in soil science. But I have heard that the secondary metabolites, the compounds inside the trichome head, while you mentioned degrade, they also change the chemical constituents of them. So you will get a different effect. So one could say that while the trichome is technically degrading as it turns amber, I'm going to get different chemical compounds in there that are being produced that will give me an effect that I may prefer over purely milky or clear trichome heads.
Yeah, like, okay, is THC being degraded to CBN? But okay, that's what I want. I want something to help me to sleep. That's that's my goal. So okay, I will delay my harvest. Again, I'm not stating that. I'm just saying that if it could be something like that you're talking about.
Yeah, that'd be an example that
Yes.
no one should take as the truth. It was just a hypothetical example. Yes.
Like, yeah, okay. A potential, circumstance. Yeah. And as you mentioned, like the publication that I mentioned about the trichome density counting is actually it was just published. I forgot to send to you. It's called Bracts, Buds, and Biases.
Oh, I have it. I do have it.
Oh, you got it?
And that's the one. I will put links to these on the podcast page for folks who want to go and check them out themselves. Yeah, because these are publicly available, which is really, really nice. I love open access stuff. Well, let's change direction. I'd be remiss not to ask you about your work now with jasmonic acid or methyl jasmonate. This is something that I heard about, oh gosh, like 15 years ago. There was a product called Jazz Spray that was a jasmonic acid product for roses that people were spraying on roses. And there was a lot of folks that wanted to experiment with it with cannabis because it's a naturally occurring chemical that we find in a variety of different plants. I think jasmine being one of the higher level plants that has higher levels of it, which is why it's called jasmonic acid. Correct me on any of this if I'm wrong, but it works sort of like it's a plant hormone essentially. And so it impacts the growth of the plant and the way it, I guess, allocates its resources. You did multiple studies with jasmonic acid. What are sort of your findings, and how does it impact cannabis?
Okay, good. I mean, I'm fascinated about that too, because basically, when we're applying like exogenous jasmonic acid in plants, you're pretty much fooling the plant. You're like, as we're talking about stress, when the plant is naturally under stress, the plant will produce, this volatile compound, jasmonic acid or jasmonate or methyl jasmonate. And then, but when we exogenously apply, you're fooling the plant. You're telling the plant that the plant is under stress without being under stress. So
when you, just to clarify for listeners, when you say like in exogenous, that means it's a external application of this plant hormone, whereas endogenous is one that's produced inside the plant itself by the plant. So essentially the plant thinks it's producing it, but you're adding it from outside.
Artificially, exactly.
Yes, artificial. I just want to clarify for listeners who may not be familiar with all this terminology because it's confusing.
Yeah, exactly. No, because I'm pretty sure, like, for example, that study that we just talked about, the water deficit stress. If we measure jasmonate, release, the plants under stress will be releasing much more jasmonate, than the control. Yeah. So the plant naturally is producing that. So our hypothesis was like, okay, why not instead of promoting a controlled stress or stimuli, instead doing that, we can just pretend or fool the plant as they are under stress without being under stress. okay, with that, so that was the idea, and we thought that it was very promising and really cool to test. But again, as everything else in cannabis, I mean, we have, jasmonic acid and jasmonates are being tested in other plants for for a long time, but for cannabis, it's very new. And so for other plants, we have much more background information in terms of when to apply, the rates, and in everything else. And for cannabis, we're just like, okay, how much should we apply? What is the optimal rate? So then we read some studies, addressing other plants, and they're like, okay, maybe this range sounds okay. So okay, let's try that, and let's create a protocol of when we apply. So the results came back not as promising as we thought. Again, I have to read those papers again. It's it's just so many other papers, but it increased trichome density, but the cannabinoid was not a statistical. The difference, it did not increase in a statistically significant level. So, but again, I think the answer, or general answer, is like we need to refine the best rate, because again, when you're talking about rate, we're talking about how much you stress the plant, and also timing. So I would assume that for these specific studies that we address, methyl jasmonate, uh, perhaps it was not the optimal application rate.
So, just looking at this first one that you did with ammonium bicarbonate, the methyl jasmonate treatment led to a significant 32% increase in trichome density. However, it did not significantly alter CBD or THC concentrations. So this supports your earlier statement saying that the increasing trichome density does not increase overall THC levels, or secondary metabolites. But this would say you had a frostier looking flower that may have had greater bag appeal, correct? Is
Yeah,
that safe to
yeah,
say?
yeah. I mean, it could be a stimulus just to force the plant to create more X structures or more protective X structures, but maybe what is inside the globular head was not as concentrated, So the higher number was not equal to higher cannabinoid concentration.
Yeah, I had a conversation with some folks in the cannabis department over at Cornell, and essentially they talked about this idea of a dilution effect. Like a plant can only produce so many cannabinoids. Genetically, it has just this genetic potential, and once we maximize that potential, now we're diluting it. So if you picture, let's just say a 3-foot plant and then a 6-foot plant that are both the same genetic cultivar. They're clones of each other. That 6-foot plant and that 3-foot plant may have the exact same total cannabinoid level because that 6-foot plant may have just spread out the cannabinoids within the trichomes over a greater surface area. It just essentially diluting it versus the 3-foot plant that might be more concentrated but have the same levels. Does that sound like a reasonable explanation, or how would you explain that?
100%. And I use that a lot in my papers, because I'm actually seeing many papers actually from Dr. Nirit Bernstein that she used this dilution effect. And the dilution effect is pretty much what you said, but I also would add that even biomass, if you're producing biomass or more biomass, this dilution effect can occur, because you're increasing biomass, and maybe the trichomes are being more sparse, farther apart. Another thing that can also affect dilution effect is water depth stress, because when the plant is under stress, it starts shrinking. All the surface area of the plant starts shrinking, and then the trichomes might be closer, apart. and this will increase the trichome density, and then maybe a healthier plant, or plant that is not going under water depth stress, the surface will be like a normal surface, and this dilution effect won't happen. So again, I'm seeing a lot that, and I'm using a lot of of this term to explain some of our results.
Yeah, that's really interesting. And I want to clarify something else too. Like the example that I gave was sort of a linear example, and the idea that these two plants are producing the they're both maximizing their genetic potential for cannabinoid production. And I think in reality, that's rarely the case. So I think in the average facility, or the average cannabis plant out in the wild, increasing yield will also increase cannabinoid production, overall cannabinoid biomass. It's just when you get to further and further along, there is a point where it's going to maximize. But I think we rarely get there in most facilities, just in real world practice.
Mm-hmm. And this also happens with other plants too, like the genetic potential of the plant. Like for corn or soybean, you can be growing the plant, even outdoors, and it's the optimal condition, perfect rainfall, perfect soil texture, perfect fertility, and the plant is not going to go up that with genetic potential. That's the max, and that's it.
Yeah, that's a good point. So I'm looking at this other study that you have, the role of jasmonates in modulating growth, trichome density, and cannabinoid accumulation in Cannabis sativa. It's a little different than the other study. Did you want to talk to me about that, or do you want me to share a little bit from the abstract?
I mean, if you
Since I know you don't have it in front of you, I'm kind of cheating
yeah,
here.
yeah.
So in the abstract, you say that jasmonates have emerged as a prominent elicitor for enhancing trichome development in cannabinoid production in Cannabis sativa. Evidence suggests that moderate methyl jasmonate concentrations can effectively boost trichome density and enhance key biosynthetic enzyme activity. I'm paraphrasing here. Ultimately, increasing THC and CBD content. However, excessively high levels produce the risk of excessive vegetative stunting. So that's really to timing. And interaction with hormones like gibberellin, salicylic acid, and ethylene further shapes the plant's stress yield, stress responses, and secondary metabolism. Do you want to explain that?
Yeah, yeah. Let's go back. That goes back to the optimal rate. So we found that, and there is it can be a wide range, but we found in other plants, there's also in other plants that 50 to 100 micromoles are somehow working for some genetics. So in some other plants, but again, it goes back to the genetic specific results. So but one thing that is very clear and very well known in the literature is when you apply too much methyl jasmonate or jasmonate, you create this stunting effect. So you really decrease the biomass. So that's why some people even use that to actually make more compact plants, produce more compact plants. So and again, that also goes back to what we talked in the very beginning of this conversation: biomass and plant secondary metabolite production. It's a balance, not lose too much biomass, and but also create a trigger that can increase secondary metabolites.
Is jasmonic acid totally safe for human consumption? And are there organic sources of this for people who are organic growers that wanted to play around with it?
I don't think there is any issue with the application of jasmonic acid or methyl jasmonate.
Okay. I haven't looked into it too much myself, so I was just curious. I know with some of these plant growth hormones and regulators, there can be. But yeah, okay, and
I
maybe
mean,
that's not something you've
it's a plant
researched.
hormone. Methyl jasmonates is an organic compound. And I guess if it's organic label, if the product that you're saying is organic label, I would assume that is not, there won't
Approved
have any
for
issues,
use in
for
organic
application.
production.
And of course,
Okay,
it changed from one state to another,
sure,
because it's
sure.
changing, across the board. But it's and again, and that's why we decide to test it, because it could be alternative for producers to use it as a potential yield increase.
Okay, interesting. Yeah, I haven't spent a lot of time looking into it, so just curious. Okay, well, last topic. I know we've been on here for a little while, but one of the things that you brought up in email that I thought we should talk about was terminology. What is the proper terminology for the plants that we see, in cannabis? Because traditionally, the term strain has been really popular for describing different cannabis. What I would say are cultivars. We know that we have different definitions for all of these things. Do you want to talk a bit about that, or do you want me to give? Actually, let me give a quick cultivar,
Yeah, I
a
want
quick
you to
overview.
start. I wanted you to start. I want to learn what is your opinion about that.
So when we talk about, proper terminology here, or classification, we have species, variety, cultivar, and strain as all potential names here. Like as humans, we're species. When we talk about varieties, for example, varieties are naturally occurring within a species. So for me, variety doesn't really apply to cannabis because it's not naturally occurring. Cannabis is obviously a naturally occurring, but we have cultivated. So a cultivar being a cultivated variety, that's something that's created and maintained through human intervention, I think really fits for the majority of what we see in the cannabis industry nowadays. So I've been using the term cultivar for a really long time. And think that is, and then strain, like I said, we've used it for years. I don't really think it's accurate. It more applies to viruses and a genetic variant within or subtype within a cultivar or within a species. So it is complicated, but I don't know what's the academic world have to say about it.
Well, so I agree with you. I mean, for all the papers that we are writing, we're using the term cultivar, because cultivar, it means that it's being bred. It's the human, it's interacting and changing the traits, and you have there's like an end goal. There are some
Yeah.
specific traits that you're looking for when you buy a specific cultivar. But some people say that and again, doing this meeting with some Cornell folks, again, I thought that I was saying something that was uh, already agreed,
Commonly accepted. Yeah.
commonly accepted, and like this is not going to be controversial. And they're like, well, Jose, what are you talking about? Then I was like, well, I mean, it's not going to be a variety because variety is something that is natural occurring when it's not being bred, and there's not really any human action to
Yep.
create a specific trait. And strain, I was like, well, strain, it's you can. And then some of them like, no, you can actually use the term strain for plants. There are some strain of rice, and then I was like, whoa, and then during that meeting, I was actually inviting someone for our cannabis symposium here that is going to be after your conference in late September. And there was like, I think this is a perfect topic to bring up because I know, especially here at Southern Illinois University, there are some people very vocal about the use of cultivar. But when they start discussing and not really agree with me, I was like, well, I guess there are some different mindsets out there, and uh, and again, I was trying to defend my opinion about using the term cultivar,
Yeah.
but it was interesting to discuss with them that some people don't really agree with that, and they have different opinions about it. So uh,
Yeah, I think that's really interesting. At the end of the day, I mean, does it matter? Probably not. But I think that it would be good to see some consistency across scientific literature at the very least.
yes,
Like, it would be good for the academic community to agree on it. Whether or not the average grower or cannabis user calls it a strain or a cultivar probably does not matter. But I think ultimately, I think like a really good taxonomist outside the cannabis industry would be the best person to ask about that and get some sort of consensus around it. But
and
so
the
we
thing
can
is,
narrow it down to two, right?
they're defending their idea about not using the term cultivar because they said that in order to use the term uh, cultivar, it need to be registered in the International Code of Nomenclature
Yeah,
for cultivated plants, the ICNCP.
I see.
You
Okay.
know, and then they're like, oh, if it's not in this, the code is not registered in this organization or institution, is not a cultivar. But I was like, well, but the thing in my opinion is more than that is the goal. Like you're breeding something with some specific traits that you want the consumer to have. So you know it's not, because if you're saying that it's not a cultivar, so then it's nothing. So then it means that wherever you're buying, there is not really consistency at all. Of course, we know that there is not much consistency. We know that if you test the blue dream from here, Illinois, and from Colorado, the profile, the cannabinoid profile will change. But at least when you're buying blue dream, that's what you buy. That's what at least you're trying to buy.
Yeah.
So then I guess to minimize that conversation and to kind of find like a middle ground, I was like, how about use the term line instead, cultivar or variety or strain. How about use like line, line with these specific traits, and then you cite the name of that specific line, because then you're not claiming anything. You're just saying that it's a line. So they're like, oh, it might be a better term because then you're not claiming too much. So
So essentially, the International Cultivation Registry Authority is responsible for the registration of all cultivars. So they're saying if something's not registered there, then it's not truly a cultivar. I guess so. I don't know. I don't know. I would agree with you on this one. Strains not typically used to describe plants. As I'm reading here, also it says it does sometimes show up in breeding, but mostly as it relates to genetic modification. So if genes of the wheat plant are altered, the offspring of that modified plant might be deemed a strain, and that makes sense to me. But this level of just intentional breeding or selection by humans on a plant traditionally in the horticulture world is called a cultivar.
cultivar,
So
I agree.
we can rule out species, we could rule out variety. Those definitely don't fit. But if you want to keep calling it a strain, there are some folks out there in the academic world that sounds like would agree with you. If you want to keep calling it a cultivar, join our club. That would work too. And hopefully someday we'll have some consensus.
or just say line that you're gonna be avoiding any confrontation because
There
it's
you go.
nothing.
Yeah. Yeah.
Because the thing is, for the controversial, like cannabis sativa being a species or subspecies, or the ruderalis and the indica, all that controversial thing, I would say that now we all agree that cannabis sativa is a one species, and we are
Yeah.
not even debating more. But this cultivar variety and strain, I guess, is something that is still being debated.
Yeah.
So
Yeah. Yeah. Awesome. Well, I enjoyed the conversation today. I can't wait to see you at the conference. I'm really excited for all the wonderful people that are going to be attending. And yeah, looking forward to it. Thanks for your time today.
Yes, thank you. Thank you. Appreciate it. It was awesome to talk to you. And, I'm looking forward to meet you in Montana.